Thanks, Antoinette. Hi, good day. Thanks, everyone, for joining us in this session. My name is David, and I'm a research fellow at the University of Nottingham's Future Food Beacon. Today, I'll be talking about some of the cadmium research we've been doing in collaboration with the Cocoa Research Center. So just recapping some of what the previous presenters would have talked about. Essentially, one of the main issues the chocolate industry faces right now is cadmium. Cadmium, it's a heavy metal found in most volcanic soils, but human activities can increase soil cadmium levels. When we eat foods with cadmium, it tends to accumulate in our body, and this is linked to a number of diseases. Similar to other plants, cacao can absorb cadmium from the soil, and it can accumulate in the roots, shoots, and beans. And thus, it's a food safety issue. Also, in 2019, the EU implemented maximum allowable limits for cadmium in chocolates. And this has affected the export of beans into Europe, impacting cocoa farmers as well as chocolate producers. So the problem with elevated cadmium, however, it's mainly an issue with beans from Latin America and the Caribbean. So there are several strategies being looked at to mitigate the accumulation of cadmium in cocoa. And these include the use of soil additives, cultural practices, and processing. However, their effectiveness dependent on the soil type, agroecology, and it can be quite costly. Exploiting genetic differences for cadmium uptake between varieties is another strategy we can explore for mitigating cadmium accumulation. And this has been demonstrated in several species. So the absorption and transport of cadmium into plants have been studied in other species. And differential uptake and storage of cadmium between varieties have been found. Essentially, a number of proteins, these are transporters, have been linked to the movement of cadmium into the plant and throughout the plant. In an earlier study involving about 100 cocoa varieties, it was found that there were differences for cadmium uptake across these cocoa varieties. The study also showed that there were differences in the partitioning of cadmium between the different plant organs. And from that study, the researchers hypothesized that there may be multiple control points in the movement of cadmium from the soil into the roots, to shoots, and eventually to the beans. Our overall goals of this project are to develop genetic markers that are linked to low bean cadmium, and then to use these markers to rapidly screen varieties that can be used as rootstocks in grafting. And this would be a more short-term goal. And then also to use these markers to support the breeding of low bean cadmium varieties with other favorable traits. And this is more of a long-term goal. So to do this, we use the genome-wide association approach to identify the genetic markers and for gene discovery. Now, the International Cocoa Genebank in Trinidad is one of the largest and most diverse public collections of cacao germplasm, with over 2,300 varieties present. The collection also has numerous varieties representing the different cacao genetic groups, as well as a number of of the different hybrid groups, like the Trinitarios and the Refractarios. For each accession, there are up to 16 trees, and then the trees are mainly propagated as rooted cuttings. The site also has moderate levels of cadmium in the soil. So therefore, the gene bank, it's a good location for screening varieties, screening cocoa varieties for differences in cadmium uptake. So in our study, we screened around 500 different cacao varieties, and this was done at the gene bank. And we screened for differences in cadmium. The cocoa varieties were also selected to represent the different populations, right? So the different genetic groups of cacao. We first started by collecting bean samples from over 500 varieties, right? And the bean samples, we separated them into the cotyledon and testa, so they were analyzed separately. Also, we harvested leaf samples, so we harvested leaves from each variety. And in fact, we would have taken those leaves from the same trees that we would have collected the bean samples, right? So the leaf samples were collected from the same trees that we collected the bean samples for analysis. So to measure the cadmium concentrations, we took the leaf samples and bean samples and then digested them and then analyzed them using ICP-MS. And this analysis was done at our ionomics facility at Nottingham. Also, for the DNA, we first extracted DNA from the different varieties and then sequenced the genomes of the varieties using low coverage sequencing. And this was also done at our sequencing center in Nottingham. So in the end, we had both the data and the genetic data. So with ICP-MS, this technique, we were able to measure the cadmium concentrations across the varieties, but not only cadmium, but we were also able to measure 22 other elements. So we got the full elemental profile across the samples, right? So we analyzed the levels in leaf, cotyledon, and testa across the different varieties, and we got up to 23 elements. And we saw significant differences across the varieties for most of the elements, including cadmium. So with regards to the cadmium, we saw that there was a wide range in cadmium concentrations between varieties in both in the leaves, in the cotyledon, and as well as the testa. So some varieties had low concentrations of cadmium, while other varieties had elevated levels of cadmium, with more than a 10-fold difference between the lowest and highest concentrations of cadmium across the varieties. And we saw this trend in the three tissues, both in the leaf, the cotyledon, and in the testa. We also found that there were significant differences in cadmium concentrations between the different fractions analyzed. So, for instance, the cadmium concentrations were highest in the leaf, followed by the cotyledon, and was lowest in the testa fractions. Overall, we saw strong correlations between leaf cadmium concentration and bean cadmium concentration. So this was good because, so besides cadmium, we generated data for 22 other elements from the leaves and beans. And we also saw strong correlations with the other elements. So for instance, we saw correlations between rubidium and potassium, as well as correlations between strontium and calcium, which we expected to see, as these are usually correlated together in other plants, as they are chemical analogs. So this gave us additional confidence in our data. Essentially though, we have a very large data set of more than 23 elements from the leaves and beans for over 500 cacao varieties, which we can explore further. With regards to the DNA, so we sequenced the genome of 502 of the accessions, right? So we did low coverage sequencing. So the coverage ranged between 1.1 to 5.4 with a mean of 3.9x. And then the reads were mapped to the reference Criollo genome. We were able to identify over 18 million SNPs, and this was before filtering. After filtering, we ended up with approximately 2.7 million SNPs of good quality, which we can use for the downstream analysis. So the first thing we wanted to do was to test our GWAS pipeline. And to do this, we selected the pod color phenotype. Now, Mortimer in 2013 suggested that pod color was controlled by a MIB transcription factor on chromosome 4. So we used our genetic data and did an association test with the pod color phenotype, and the pod color phenotype being anthocyanin present, this is absent. And when we did that analysis, what we got was a massive peak of SNPs on chromosome 4 associated with pod color, right? So we got this massive peak of SNPs on chromosome 4. And in fact, this peak of SNPs, it was over the MIB transcription factor identified by the 2013 study. Additionally, while the 2013 study had resolved the pod color to a broad peak of SNPs, with the most significant SNPs spanning about a 1 megabase pair range, with our data set and our pipeline, our top SNPs had a very narrow range, only spanning 0.1 megabase pairs, which indicated our pipeline had a higher resolution in identifying genes of interest, which was good. So having tested our GWAS, we then performed our GWAS mapping analysis with the iNome data. And this would have been the data that we collected from the gene bank across the 500 varieties. And what we saw was that for most of the elements, we saw strong associations across the different tissues, which was good. With regards to cadmium, the associations were not as strong compared to the other ions, but still the analysis identified multiple QTLs for the leaf, cotyledon, and testa that may control cadmium in cocoa. And in fact, some of the QTLs were common to all three tissues. So then we looked at the genes linked to each QTL and their potential role in cadmium accumulation. And then afterwards, we then selected our top SNPs. So the top QTLs. So the selection of the QTLs were based on selecting those SNPs, the associated SNPs that had the high minus log P values. Also, we selected the QTLs that had the largest effector size, and also those that had differences in cadmium between the alternate alleles. So based on that, we were able to select our top QTLs for further analysis. So right now, we are currently validating these QTLs that we have identified from the GWAS study. And we are performing a number of experiments, such as using a biparental process. And this work is currently ongoing. So overall, our pipeline is clearly working, right? We are currently developing our GWAS pipeline into a flexible platform that can now be used for analyzing other cocoa phenotypes, which we will be doing in the near future. I should say that this project has been a team effort, right? It's a team of us who has been working on this, both at the Cocoa Research Center and at the University of Nottingham. And we are very grateful to Mars Wrigley for supporting this research. So with that, I thank you for listening, and we're open for questions. Thank you.